Okay, let's see. Set up- Okay, hi everyone. This is the cell process um, review for your bio exam. Hi. If you have questions, just go ahead and start asking them. Otherwise, I do have some questions that my students gave me, so I'll just start on those if no one has questions. Okay. I don't see any questions yet. Sorry, I'm going to close the door there. Oh, I don't. Okay, hold on a second. All right, so let's start with. Okay, so Sindhu, I see you say you don't understand cellular respiration. Um, can you clarify what part of it you don't understand? And while you are doing that, I'm going to answer Arshia's question. Can you please explain the cell cycle with the chromosomes and chromatids and all? Okay, so the big picture, let me see if I can find something to use as my visual aids here. Let's see. Oh, he's pretty close. Okay, so remember your. Okay, I will get to your cellular respiration questions in just a second. So remember the idea with um, chromosomes in the cell cycle is assuming that your cell is going to divide, right? Then in order to split something in half and still have enough for both new cells, you have to first double your DNA, right? So first you have interphase where the cell gets bigger and the DNA is going to duplicate or replicate. So imagining like this pencil was like one chromosome, right? And then during interphase, what will happen is in addition to just making the cell bigger, it's also going to duplicate the chromosome. So now you have two. Okay, sorry, pretend those are identical. I know they're not, but pretty close. Yes, don't worry. All the questions you guys need, I will get in a second. Let me do these first, okay? So after your cell cycle, you're basically going to have, instead of 46 of these, right, single chromosomes, every chromosome looks like this. Every chromosome is made up of two sister chromatids. These are like identical twins, and they're stuck together temporarily, right? Now, with that much DNA, you really have to have a really organized way of separating things out. So then what you're going to do is now during mitosis, you need to divide that up very evenly, right? First, during prophase, the chromosomes actually condense. They basically get more compacted, so they're easier to split apart. Because if you just had like these big stringy pieces of DNA, it'd be really hard to do that. So the 46 chromosomes, each chromosome condenses into what we call chromosome. Before that, it's called chromatin, which is uncondensed DNA, right? Then during metaphase, they line up all along the middle. So imagine if I had like multiple, let me see if I can find more of my pencils here, right? So let's say I had more than one chromosome, right? They would line up along the middle of the cell. And then in anaphase, they're going to pull apart. So they won't, I don't have enough hands to show you, but the idea is that each set of sister chromatids, right, is going to um, separate. Oh, I do see a question that's relevant here. It says, um, so chromatids and chromatin. So chromatin is uncondensed DNA, and then chromatids are the two identical copies of it. We call them sister chromatids. Okay. So in metaphase, they end up in the middle, like single file. Anaphase, they pull apart. Telophase, they keep going, and then you split the cell fully, and then you now have two cells instead of one. But now each cell has all of the DNA that the original cell had. So now you've basically made a copy of your original cell. Okay. Let me go back up and try to get all the other questions you guys had. Okay, so let's start with glycolysis. Sindhu says, I don't understand how NADH is created in glycolysis. So NADH is think of it as like a full electron carrier, right? And so it's constantly being um, filled with electrons and then removing the electrons. So you're constantly having that molecule, which is NAD plus, have electrons added to it, becomes NADH, right? And when it drops off its electrons, it regenerates the NAD plus. So glycolysis is basically a bunch of chemical reactions. And some of those chemical reactions, part of what they do is they take electrons from the glucose and move them from the glucose to the NAD plus and therefore make NADH. Okay, it says, how are carbohydrates and cellular respiration related? They are very, very closely related. So think about what you put into cellular respiration, right? At least the way we've mainly talked about it is glucose. Glucose is a carbohydrate. So the first main relationship between carbohydrates and cellular respiration is the carbohydrates are really what you're putting into cellular respiration to make it happen, right? I mean, that's really sort of the big thing. And that can be true of any carbohydrate. Even though we talk about glucose, anything else can basically turned into something that's either like that's going to be, you can turn it into something that's like glucose, or you can turn it into something that's just under that, like, the next step down of what glucose becomes in that whole process. Um, okay, Minsu says, can you explain how plants can make guard cells open and close? So remember, the guard cells are like the lips of the stomata, right? In order for eh, gases to get in and out of your stomata, you have to have your guard cells opened or closed. And so the plant can then use osmosis and active transport to do that. If it wants to open the stomata, it means you want to fill the guard cells with water. And so it does that by first pumping in the potassium ions, the K plus. After you do that, then water follows by osmosis, because now you've created um, the inside of the guard cells have so much K plus and not so much water, that water wants to flow in from high concentration outside to low concentration um, inside, right? And when it does that, it's like putting water in a water balloon. It stretches, and instead of being closed, the guard cells basically plump and they pull open, and now there's an opening where the gases can come in and out. Now, same thing if it's like too hot, there's too much water being lost from the plant, and the plant now wants to stop all that water loss, it can do the opposite. It can pump K plus out. When it pumps the K plus out, again, now it's making the outside of the outside of the guard cells more hypertonic, right? Water will now flow out of the guard cells and into the surrounding area. And then it's like a flattened water balloon, and it slams shut. Okay, Gita says, how are the processes of photosynthesis and cell respiration connected with the seasonal fluctuations in atmospheric concentration of CO2 and climate change, and how do they affect photosynthesis? Okay, let me unpack that question. Well, the idea is that you're going to have various levels of CO2, right, throughout the year based on various, seasonal things that might be happening depending on the plants and such and the animals. Um, and so ultimately, how much CO2 is available does play into then how much photosynthesis can happen, right? To an extent. I mean, it can also be that there's so much CO2 the plants simply cannot absorb it enough, right? But they're all related because CO2 needs to go into a plant in order for glucose to be produced, right? And then once glucose is produced, then cellular respiration uses that glucose for energy. Um, okay, I never really got when the stomata open and close when it's windy, dry, or hot outside. Can transpiration increase even if the stomata closed? So no, first of all, transpiration won't increase if the stomata are closed. Think of it as the plant is going to respond to the situation, right? Meaning, if it's really windy outside and you keep blowing the air away from the water specifically, not just the air, like the wind is blowing the water around the plant away, then now water is going to evaporate more out of the stomata, right? But plants, their stress level is essentially tied to how much water they're losing. Like they don't want to lose too much water. So if a plant starts to get the signals that it's losing too much water and is now at risk of drying out, which is going to be really bad for that plant, then it's going to start um, causing certain things to happen in that plant that we're going to close the stomata so that it won't lose too much. So really, plants react by saying, I don't want to lose so much water. I'm now going to start doing things to stop myself from losing too much water. So that's usually when they're going to close their stomata, right? It's the same thing when it's dry. Like it's dry outside, more water is evaporating. It's hot outside, water evaporates faster when it's hotter. So all those things are going to make the stomata close. Whereas if you have a situation where it's just kind of very humid outside, and there's already a lot of water out there, then you don't really have to worry as much about losing water, right? Because there's already plenty of water outside. There's water's not going to leave as quickly, so the stomata are more likely to just stay open. Um, How are stem cells and cancer cells similar? Well, um, they both are similar in the sense that they have activated telomerase, so they don't go through just a few divisions and then self-destruct. But other than that, they're not that similar. I mean, that's their main similarity, I would say. Right? Stem cells are like the precursor cells that are going to become a lot of other cells, so they're kind of just waiting around for their turn or chance, I guess you could say. Whereas the cancer cell is like a cell that's like a rebel cell. It's not listening to instructions anymore. Um, okay, how is 4-ATP created during glycolysis? I don't know how many details you need to know about that in terms of specifics. So I'll keep it fairly general. Basically, remember, glycolysis is a bunch of chemical reactions that transform the glucose into two pyruvic acids. And so, in the process of doing that, it's like origami. It's like each little step is very specific, right? So, sometimes some of those steps, while you are making like one intermediate, one like middle molecule to another one before you get to the um, pyruvic acid, sometimes one of the things they'll do is they will, as a process of that reaction happening, they'll actually take an ADP and add a phosphate to it, right? And that happens essentially with four ADPs turning into four ATPs. Glycolysis, remember, is the first process of cellular respiration, which is where you are going to um, make the glucose, which is too large to get into the mitochondria, into a two 3 carbon molecules called pyruvic acid, which can then enter the mitochondria. Um, okay. Uh, let's see. Sorry, I'm trying to make sure I don't. Oh, I missed a lot of questions. Okay, hold on. You guys are asking questions really fast, which is okay. Let me see. Um, Why do plants that are growing near highways with a lot of car exhaust grow faster than plants that grow in other areas? Well, car exhaust has a lot of CO2, right? So if you have a lot of CO2, then theoretically that CO2 could be used by the plant to make glucose. That glucose gives it energy, and then theoretically the plant could grow more, I suppose. Um, okay, yes, thank you. The difference between chromatin, chromatids, and chromosomes. Okay, so it's really all vocabulary that has to do with DNA. Okay, um, chromatin versus chromosomes is referring to the two different ways that DNA can exist. It's still DNA, it's still that same biomolecule, but one way of thinking of it would be like, let me see if I have something here. Sorry, my visual aids are limited here on my desk. Hmm. Okay, we're gonna use this hairnet or something. Okay, so think of the chromatin like this big stringy hairnet. Okay, like it's all spread out and it can be used for, all the stuff the cell's going to do, right? But when you have that much DNA and you're getting ready to divide it, you don't want to have all these big stringy chromosomes like running, just floating around. So what the cell starts to do at the beginning of the cell of mitosis is it's basically going to make it more compact. It basically condenses it, right? So imagine taking that and just sort of twisting it up and making it really, really tight. So it's like a little ball of DNA, much more compact, I mean, a lot easier to split, right? Compared this to this, it's a lot easier to like divide things up when they're all compacted. Okay. Now, chromatids are referring to sister chromatids, which is to say, during interphase, when you copy the DNA so you have enough to split to the two new cells, you're going to have two identical copies. Those are the chromatids. Um, let's see. Yes, overall, so with glycolysis, it's one of those things where there is some ATP spent. There's also some ATP used because someone was saying, I think two ATPs are created. So you basically make four ATPs in the process of glycolysis. But you also use two ATPs to sort of some of the reactions require that. So the net gain for the cell is it's still going to make two ATPs overall in glycolysis, which is not a lot, but it's something. Um, Matthew's asking, how do environmental conditions affect photosynthesis? Well, again, environmental conditions means things like wind, humidity, heat, right? Sunlight, all of those things are environmental conditions, right? So if there's enough sun, you can do more photosynthesis, right? If there's not as much sun, you can't do as much photosynthesis. If it's very windy and you have to start closing the stomata, well, not only is the plant not going to lose so much water, which is a good thing for the plant, but it also means you've closed the window that allows CO2 to come in. So now you're also going to be limiting how much photosynthesis can happen in that sense. Svenja says, why is it important for chromosomes to condense and separate from each other before they are divided? Um, they don't actually separate from each other before they're divided. I mean, it's the condensing is important, right? Because again, think of like if you had to separate a bunch of things which could get really tangled because they're all spread out versus a very, very compact little ball, it's just going to be easier to divide it up. Think of it as you have a huge basket full of yarn and it's like all different colors and all different pieces, right? And it'd be really hard to like split that evenly into two groups. But if all of the yarn was each one balled up very neatly into a ball, then it'd be easy for you to be like, okay, here's two white yarns, two blue yarns, two purples. I'm gonna split one to each side, and that way I'm done. Chlorophyll is the molecule inside the chloroplasts in the thylakoid discs. It's the thing that's going to have an electron in it. So when the sunlight shines on it, it excites that electron and then it excites it and it starts the process of photosynthesis and the light reactions. Okay, so Sahana's asking, how do stomates open and close? Again, that's the potassium, right? It's all about potassium and water following. So you want to close if you want to open the stomata, you pump potassium in, water follows, right? Think of it as like, again, it's like a water balloon. It's full now. And now the stomata, which before the guard cells were like this, now they open up and now there's an opening for gases to get in and out, as well as water. Whereas if you want to close the stomata, again, pumps potassium out, water follows by osmosis, they slam shut, no more hole for the gases to come out. So guard cells are really like the lips. They're keeping the gases out if they're closed, or if they're apart, they're letting gases through. The main differences between spongy and palisade mesophyll cells is just the idea that they're both photosynthetic cells. They can both do photosynthesis, but their location is different. So palisade mesophyll cells are at the top of the leaf, and then spongy mesophyll are usually below that. Spongy mesophyll are a little bit more spread out. There's more air spaces between them. But because the palisade mesophyll cells are at the top, they're the ones that the sunlight's going to shine on and actually be able to, excite the electrons in those first. So they tend to do more photosynthesis. How does the phloem play a role in photosynthesis or cellular fission? It doesn't play a role in photosynthesis or cellular fission, but it actually does play a role in what comes before that or after that, right? In the sense that the phloem is for transporting sugar, right? So every cell in the plant is going to do cellular respiration. Only the photosynthetic ones that have chlorophyll are going to do photosynthesis. So one way you might think of it is you might make the glucose in photosynthesis in like a leaf cell, right? But then maybe it needs to go down to the roots where there isn't photosynthesis happening. So the phloem is how the sugar that's made in one part of the plant gets transported to another part of the plant where it can then use it to do cellular respiration. Let's see. I have different notes for what happens in the phases of a cell. I have ones. Okay. The notes shouldn't be that different. It's still the same idea. It might be the order of things is a little different, but still remember in terms of the cell cycle, Anika, I'm guessing you mean what happens in each phase, like prophase, metaphase, anaphase, telophase. So I'll just review to make sure you have it, and then we'll kind of just take it from there. Okay. So and this is in the review sheet though. So in prophase, again, your chromosomes are condensing, the nuclear envelope, the membrane around the nucleus is disappearing. The the chromosomes are condensing. I think I already said that. And then in metaphase, think M for middle, they line up along the middle of the cell, right? They sort of line at single file. Each one, again, is the two sister chromatids, right? In anaphase, think A away, they get pulled apart. And then in telophase, they continue to get pulled apart until now a new wall basically forms between them where they pinch off, and now you have a new cell, one on either side. Mitochondria and chloroplasts, and then that's the end of the cell cycle. Mitochondria and chloroplasts are related in different, so they're both organelles involved with energy processes, and they are definitely very related, right? The chloroplasts are the ones that are going to capture the sunlight's energy, transform it into glucose, and then the mitochondria are going to be able to take that glucose and turn it into ATP, which the cells can use. Um, what form does energy take throughout photosynthesis and cell respiration? It takes a lot of different forms, right? I mean, it starts as light energy, then it's converted from light energy into the chemical energy that's in electrons, and then from the electrons, it's transferred to the glucose, and from the glucose, then it's eventually, if you go through cellular respiration, it's going to be made into ATP. So cellular respiration is basically the exchange of air. No, so cellular respiration is the process of taking the glucose and turning it into ATP. Now, in order to do that, you do require some gases, but that actually doesn't have to happen from the stomata. Because remember, like humans, we don't have stomata, and we can't do, but we can still do photosynthesis, right? So cellular respiration is really that process of making the ATP, which is the chemical form of energy that your cells need to use from glucose. How is cellulose related to photosynthesis? Remember, cellulose is a polysaccharide. So it is what you string together glucose to make cellulose. And so it's related to photosynthesis because it's sort of something that happens after photosynthesis. If you make a bunch of glucose in the cells are going to use some of that glucose to make their cell wall, which is made of cellulose. What fermentation is? Fermentation, remember, is the idea that you can't do all of cellular respiration, right? You don't have oxygen. You have then, because of that, and there's no final electron acceptor, the ETC shuts down. If the ETC shuts down, the Krebs cycle shuts down. So now all I'm left with is glycolysis. Well, glycolysis is not making me much energy, but it's still going to make me a little bit of energy. The problem is glycolysis needs to have empty NAD pluses to give electrons to in order to do glycolysis. And normally you would get those empty NAD pluses by going to the ETC, and NADH drops off its electrons, and now you have empty NAD pluses to go back to glycolysis. Well, the problem is if you're not doing the ETC, you basically have to find a different way to empty that NAD plus. And so fermentation is a different way, it's an alternate pathway to regenerate the NAD plus, to empty the NADH, remake NAD plus, so that glycolysis can happen. What's the equation for photosynthesis? I think that's in your notes and I think someone just gave it to you, so I will skip that question. And again, NAD plus is recycled in that sense, right? You recycle it in the sense that you empty the NADH to remake it so that it can be used for glycolysis. The cell plate, don't worry about that. I didn't actually talk about that in too much detail. The cell plate just is when that new wall forms in a plant cell between the two cells that are forming. And so it's literally like a little wall forms. So now you have, um, um, he says on the study guide, it says electron is picked up by blank. I don't get that question. Let me find the question. Let's see. Hmm. Anika, I'm not sure where it is, so I guess I will just adjust it generally in hopes that it answers your question. Um, basically, the electrons are picked up, depending on what we're talking about, right? If it's an electron in photosynthesis, then the excited electron gets picked up by NAD plus to become NADPH. If we are talking about cellular respiration, then those electrons get, which are high energy electrons as well, they get picked up by FAD or NAD plus to fill NAD to make NADH or FADH2. Lucy, the layers of the leaf are basically from on page 596 of your textbook. It's just looking at the different parts of the leaf, which I also gave you in a blue packet, the plant form and function. It's just, want to know, again, I don't need you to memorize the whole diagram. I want you to just be able to notice things like here are the guard cells, here are the stomata, here is the xylem, here's the phloem, here's the palisade mesophyll, here is the spongy mesophyll, etc. Okay. What I mean by plant stress levels is just like humans can get stressed, plants can get stressed out too, right? But it's different in plants. Like they don't worry about like their tests the next day. What they worry about is having enough water. So for a plant, when a plant is under stress, molecules get released that will cause it to do things like close its stomata. Let's see. I think I already explained fermentation, so hopefully people are okay with that. Okay, stem cells, Shreyas, are remember, they're the precursor cells. They're the cells in your body, which I mean, human adults don't have very many of them, but they're like the non-differentiated cells. So they're the cells that could still become a lot of other types of cells in the body. For us, mostly blood cells and maybe immune system cells. But in an embryo, there's a lot more stem cells, which could become any cell in the body. Cells would undergo mitosis for a few different reasons, right? It could be for growth. Like, for example, if you think about how small you were when you were born and how big you are now, you're much bigger. And the way you're much bigger is because you went through a bunch of mitosis. You created a bunch of new cells from your existing cells. It could also be, let's say you get a paper cut, right? Now you have a hole essentially in your skin and you need to fill in that hole. So you're going to grow new cells to fill in the hole. So those are some of the main reasons you might see mitosis happening. Can I explain mitosis? Yes, let me see, how can I abbreviate it? So remember, mitosis is technically the division of your DNA. You want it, and it's the idea of dividing your DNA equally into two new cells. And so you want to do it in a way that's really organized and very systematic, because you wouldn't want to accidentally give one cell way too much DNA and one cell not enough. So the way that you do that is that you want to have it, dividing the DNA in a very, again, systematic way. So you condense the DNA, you line it up down the middle, you split the two halves, and now you have two new cells. Okay. Let's see. What does glycolysis exactly do? I think I mentioned it before, but in case you missed that part, because I know people are joining at different times, glycolysis um, glycolysis really just takes, again, that glucose, which is a 6-carbon molecule, makes it small enough into a 3-carbon molecule to put it into the Krebs cycle. Let's see. Let me make sure. Oh, sorry. It looks like. Okay. All right. Sorry, guys. I didn't realize this many extra questions came in. Okay. Sorry. I don't want to make sure. Okay. Okay. I think we're in a place. Yes. Okay. I think everyone's answering questions too about glycolysis correctly. Yes, Shreyas. So they're waiting essentially till your cells need certain things, right? For example, your red blood cells only live 120 days. So you have bone marrow cells in your femur, in your pelvis, right? Which are undifferentiated cells. And what's happening is that as needed, as red blood cells die and you need to replace them, then you make new ones from your bone marrow. That's why if you donate blood, it's not like you're losing your blood forever. Like your body will regenerate and make you new red blood cells from those stem cells. Can I explain all of cellular respiration? Okay. So I'm gonna give you the abbreviated version. Remember, the idea of cellular respiration is to turn glucose, which is energy your cells can take in but can't do anything with into ATP, right? So in glycolysis, you're going to take the glucose and you're going to break it down into pyruvic acid, 2 pyruvate acid. So it's a 6 carbon molecule glucose into 2 3 carbon molecules pyruvic acid. And then each of those pyruvic acids will go through the Krebs cycle. The Krebs cycle is really just taking now the energy that's still in the pyruvic acid and transferring it using electrons into a bunch of NADHs and FADH2. So you're transferring all that into your electron carriers because those electron carriers are now going to go to the ETC, which is the final step. And when they do that, they are going to use the energy from the electrons and make it into ATP. That's kind of the really condensed version. Now remember, hello. Okay, mommy has to talk to them. You say hi, girls.
Sit on their lap.
Okay, you say hi for just a minute, and then mommy has to continue talking.
Hello. But can we stay here?
Just for a minute. Okay, so what you end up with is you need oxygen, right? Because those electrons, when NAD plus and FADH2 drop them off, then they actually have to give the electron at the end to something. Because if the electron just stayed in the electron transport chain, no new electrons could be dropped off. So oxygen is your final electron acceptor, and when it adds the oxygen with the electron and hydrogen, it becomes water. And mommy's talking about how energy is made in your cells.
Oh, I know.
Okay, no, let mommy talk about it. Okay, does creating ATP through the process of fermentation take longer? It's a much shorter process, so I don't know if it takes longer, but it's not going to generate as much ATP. So, how do I remember the phases of mitosis? I think there's 2 different ways you can do that. Number 1, no, thank you, Sammy. No. Okay, mama needs to talk to them. So there's 2 ways you can remember. If you want to remember the order of the phases, you can think PMAT, which is PMAT, right? So you can think of like an acronym to go with it. For example, like pay me anytime, right? Prophase, metaphase, anaphase, telophase. The other thing you could remember is like a hand model. You can use your hands. You can remember like, okay, your hands are like the chromosomes, right? So in the first part, they condense. Oh, thank you, Gina. Right? In metaphase, they line up down the middle. In anaphase, they get pulled apart. And then in telophase, you now have 2 cells. Okay. Okay, do we need to... Oh, you don't need to know about acetyl-CoA and coenzyme. I mean, just know that they're technically part of how you end up making the... how you go into the Krebs cycle, because if you are...
Just push the button.
Thank you, Sammy. Okay, I need you guys to go back outside.
No!
Okay.
I
No,
want to
thank
stay with you.
Okay. Then you need to be quiet, please. No, don't push buttons. Thank you. Okay, so with
What should we draw for that?
I don't know. Draw them a leaf, please. Okay. Oh,
I need blank paper.
over here.
Where should we draw?
Okay, so with acetyl-CoA and coenzyme A, you just want to know that it's an intermediate, right? That you're turning that pyruvic acid into... No, thank you, Sammy. If you're turning it into the Krebs cycle, you basically need to make it into acetyl-CoA first. Okay.
Should I draw it with any diagrams? Should I draw with any diagrams?
Gina, you can do what you want. Okay, it says, are the 2 types of DNA condensed or uncondensed? Yeah, it's basically, that's the 2 types of DNA, because it's really still just DNA. It's just, what form is it in? Is it all clumped together, or is it separated? Can I review the Krebs cycle? Okay, and I'll show them later, Gina. Thank you. Okay, you can show them real fast.
No, but
Quickly.
Are you guys talking about leaf?
No, just show it quickly. I need to get back to this. Okay, Gina wants to show you her leaf. Okay, there you go. Thank you. No, mommy needs to talk to them. Okay, so the Krebs cycle, again, the main purpose is really to take the energy that's still in pyruvic acid and use it to transfer it into those electrons. And when it does that, then it will then be able to eventually drop off those electrons to make ATP and the ETC. No, the 2 types of DNA are actually, again, chromatin and the condensed DNA. Okay. How does apoptosis in the brain affect us? No, thank you, Sammy. No. That's not okay.
I'm doing that.
No, please don't do that.
On my head too.
No, I need you guys to go find Daddy, please.
No.
No, that's not helping. Thank you. Oh, careful. Okay. So apoptosis, remember, is like self-destruction. If it were to happen right in the brain, then you would lose brain cells. And in the case of the brain, the cells in your brain actually don't go through much, if any, mitosis. So that becomes really problematic. Sammy, no, thank you. No, stop,
I
please.
want it to be
No,
dark.
no, thank you.
I want you to look silly.
No, thank you. So yes, you could lose, function in the area of the brain. Apoptosis, remember, my group, my students, I didn't name it that. It's the self-destruction of cells that happens when cells get either too damaged or they have divided too many times. Transpiration is the evaporation of water from the stomata, and that's what drives the pulling of water up the xylem. Go ahead and draw, please.
I just need to get a marker. I need the
Okay.
pink marker up there. Can't reach.
I'll get it for you. Thank you. Sorry, guys, extra interruptions. Okay, so the difference between the xylem and the phloem, yes, is what they're transporting. They're two different highways, but with very specific things that are moving, right? The xylem basically moves water, and then the phloem is specifically moving sugar, not just nutrients, it's sugar. But it's sugar water because you can't really move sugar without some fluid to flow it, right? To move it.
Hi, Mommy.
Hi. Oh, it's like a party in here. Okay.
There's a prank.
Okay. No, Gina. Gina, I need to talk to them right now. Thank you. Okay. Spongy mesophyll are just photosynthetic cells. They are a little lower on the leaf, so not as much photosynthesis is happening because they're not getting too much sunlight from the top. Transpiration definitely has an effect on photosynthesis because it's what allows you to pull the water into the plant. And if you didn't have water, you wouldn't have any photosynthesis happening because you wouldn't be able to do photolysis. Plants would grow slow in the desert because they have to close their stomata, right? They have to balance that idea of
I
like.
love it too.
Oh, okay. So plants would have to grow slower because they would basically end up, if they close their stomata, then they're not going to have as much CO2, which then limits their photosynthesis. So there's it's limited.
This is ready, Mama.
Okay,
this is
hold
ready.
on. Thank you, Sammy. Okay, how is energy captured, converted, and stored? So energy is captured by the chlorophyll. The chlorophyll molecules are the ones that actually absorb the sunlight energy, and then they excite those electrons. And when they excite those electrons, then they basically go with Daddy, please. No, Daddy will talk to you. Thank you.
Hey, I want to stay here.
So when they absorb that sunlight energy, now it gets put into a electron transport carrier, the NADP plus, which becomes NADPH. And it's stored temporarily in the electron carriers. It's temporary. Okay. Can I elaborate on the part of cellular? Yes. So remember, it's a chain of proteins. That's what the electron transport chain is, right? So you're dropping off the electron, it's getting passed down a chain. But at the end of the chain, you have to give that electron to some molecule. Otherwise, it becomes like a traffic jam. Because if the last protein in the electron transport chain can't drop off its electrons, then it's full. Okay, no, thank you. Right, then you can't drop off your electrons, and the next one can't drop its nuts. Like you basically imagine like you're passing something from your friend to the next friend, and you have like a chain. But if the last person stops dropping off their electron, their item, then everybody backs up behind them. So it's the same idea. So in this case, the final electron acceptor in cellular respiration is oxygen, because that electron is given to oxygen, and now it becomes water. You want to get down? No? Yes? Okay. Okay. Let's see. What are limiting factors in a jungle or desert? Really, it means things that might limit the amount of photosynthesis or cellular respiration. Probably photosynthesis, honestly, more so. Jungle's probably a little less so. There's a lot of water, there's a lot of sunlight. But for a desert, definitely there's much more. I would say Okay, greater limiting factors. You're going to have things like it's the heat is a limiting factor because again, it's not going to be able to keep its stomata open as much. It's dry, right? Things like that. The structure of a chromosome. I'm not sure what you mean by the structure of a chromosome. A chromosome is just a section of DNA. Like imagine like the Harry Potter books, right? If you look at like all the Harry Potter books together, that is like the entire genome of Harry Potter. It's all the DNA of Harry Potter, right? But it's divided up into a certain number of books. So like each book is like a chromosome where it's like a section of the DNA. And that's a chromosome. We didn't actually get more into the chromosome structure yet. So yes, stomata close basically when they're losing too much water. That's the big thing. I don't know if Mr. Gan went over chromatins and stuff, so I'm sorry, I don't actually know. That would have to be something you'd have to find out from your other classmates. Okay, photosynthesis. Sunlight, water, and CO2 makes oxygen glucose. Yes, that's correct. And then cellular respiration, oxygen and glucose, yes, gives you CO2, water, and ATP. Correct. Okay. Thylakoid discs are inside. Yes, thylakoid discs are inside the chloroplast. Oh, not right now, thank you. No, you go outside to daddy.
The door is locked.
Oh, you want mommy open it for you?
No.
Sorry, guys. Okay, you go outside to daddy then.
No.
Okay, then you have to be quiet here, okay? Okay. So thylakoid discs are inside chloroplasts. So yes, since palisade mesophyll cells have a lot of chlorophyll, a lot of chloroplasts, they're going to have thylakoid discs. P53 and cyclins, they are part of regulating the cell cycle. P53 is like your proofreader. It makes sure things are okay before it lets the next part of the cell cycle happen. And one of the things they let happen, if it's ready, is to make cyclins, which allow it to move on. Yes, Joanna, exactly right. How does a change in the surface area to volume affect tissue? Basically, think of it as like the bigger the cell is, the greater the volume and the surface area, but the volume and the surface area don't get bigger at the same rate. So it means like if you have all the stuff that needs to like come in and out, baby, no thank you, of a cell, and the only place it can get out is the cell membrane, then if your cell membrane is not big enough to support all the stuff in your cell, that's going to be a problem. So that's how it affects diffusion and active transport. You just don't have enough space to do all those things you need to do. What happens in prophase is, again, the condensing of the DNA, and then the nuclear envelope starts to disappear, and so that way you're kind of getting ready to divide things. When it is windy, humid, hot, dark outside, what does effect on weather? So again, it's a matter of, you have to think about, as a plant, I don't want to lose too much water, right? So if it's hot, that means water is evaporating really fast. It's going to eventually close the stomata so it doesn't lose too much water, right? If it's windy, it's the same idea. You're blowing away all the water around the plant, so more water is leaving to sort of compensate for that. And it's, again, going to close after a while. If it's humid, it's the opposite. So if you have humidity, that's like not really going to change things, because it's going to slow down transpiration because not as much water is leaving. Same thing with the dark. I mean, technically, it's not really going to speed it up, but it's also not going to hurt it in that sense. Okay, cyclins again. Cyclins in P53. I think
Where you leave this?
It's mommy's scarf. Do you want to wear it?
No.
Okay, I'll wear it later, okay? You don't wear it now? Okay. Okay, thank you. Okay. Can I explain osmotic pressure? Osmotic pressure is essentially just water pressure. It means when you have more water in an area, it like pushes on the stuff around it, and that's osmotic pressure.
Yeah,
so when water flows into the guard cells, then there's more osmotic pressure in there. No, thank you, baby. And that way, that's what makes the stomata open.
My color.
You want a color? Okay, here. Color here.
No, I don't have my pen.
And you have to go ask daddy. Can you go ask daddy?
I don't have my paper.
Go ask daddy.
Okay, one.
Okay. Let's see. The thing that stops cancer cells from self-destructing is the telomerase. The idea that normally your telomeres get shorter and then your cell self-destructs because now it's, the cells are getting to a point where they might start to damage the DNA that's really important before the telomeres are all gone. But if you keep adding the telomeres on, you're not going to do that, right? Okay. Let's see. When a cell doubles in volume, no, the surface area and volume don't increase at the same rate. That's the thing. So, Anita, they do not. They both increase, but the volume gets bigger faster than the surface area. Okay. How do cells become specialized? To be honest, it's something scientists are still trying to figure out. Simply put, it's a matter of a cell getting certain signals so that it starts to make certain proteins, because not all the cells in your body make the same proteins. So what scientists are still trying to figure out is how do you go from everybody has the cookbook and now you're telling each cell which recipes to cook and which proteins to make. Yes, you can say for prophase that the DNA condenses from chromatin into chromosomes. That's fine. The two types of fermentation are alcoholic fermentation and lactic acid fermentation. It occurs in different organisms, but the end result is the same. You're still remaking NAD+, so you can keep glycolysis going. Let's see. What is everything which regulates the cell cycle? I mean, the only ones we talked about were P53 and cyclins. There are other things that regulate it in a lot more detail, but I don't need you to know that. Just know that P53 is like the guardian molecule, like it's checking to make sure things are okay and correct in the DNA. If they are, then it'll let it move forward in the cell cycle. And one of the things it does is make the cyclin molecules, which are the molecules that allow you to move from one step of the cell cycle to another. Okay, hopefully the surface area to volume part I explained. The function of the telomeres, again, is it gives you sort of that buffer, right? That as your cells divide and your telomeres get shorter, it gives you something to wear away that's not actually like instructions. Shreyas, do cells self-destruct because their telomeres? Yes. I mean, well, they think that's part of it, right? The cells self-destruct because their telomeres are getting too short. And yes, it will damage the DNA. And that would be really bad. Okay, let's see. How would glucose from a grape provide energy to your muscle cells? Well, remember, glucose, no matter where it comes, whether it's from a grape, an apple, your candy, is all going to be the molecule that goes into your cells in order to generate ATP. Yes, it turns into ATP. So in fermentation, remember, electrons are constantly being moved. So you asked in fermentation, when the NADH is recycled back to NAD+. Oh, careful. Oh, no, not that pen. Thank you. Your mommy gave you this blue pencil. How about this? Okay. Thank you. No, you want a color? Here. All the colors. Okay. Yeah. Let's see. The electron basically goes to another molecule. Like, you don't have to track the exact location, but think of it as the electrons will go to another molecule as the energy is moving around. Okay. Yes, that's correct. Is there a set list of everything? You have the resources. I think it's purple that I gave you, or blue. That's where the master list of everything is. The difference between telomerase and telomeres. Telomeres are the parts on your DNA. Telomerase is the enzyme that adds the telomeres back on. Yes, essentially studies things that you've gotten from this unit and you're good to go. It could go down the ETC, yes. The electron doesn't have to be. It could be just given to another molecule. Let's see. Is your bone marrow stem cells? Yes, bone marrow is the cells, are the stem cells in adults, right? Again, in embryos, you have more stem cells, but those are where your stem cells are now. Okay, are the major events of the cell cycle, yes, are interphase and mitosis. Depending on how much detail your teacher went into, you might need to know the phases of mitosis, like my students, you do a little bit. And then, okay. How are cell division and cell death regulated? They're regulated by those molecules, really. You know, the p53, the cyclins, all of those molecules and signals coming into the cells basically help determine whether or not the cell can move forward in cell division. If it's too damaged, it gets triggered to self-destruct, etc. And if the cell cycle is not regulated properly, then essentially that's, you know, when you do have issues with mitosis cancer, right, where cells just start to divide when they're not supposed to. I think the idea is that alcohol is essentially is a toxin, and so it can damage cells and DNA and such, and therefore it could then cause mutations in the DNA, which then could, not definitely, lead to cancer. Let's see. Okay, sorry, let me make sure we're getting all these. So there's not glucose in every food you eat, first of all. Okay, actually, hold on. The questions have gone really fast. Oh my. Sorry, guys. I'm just trying to make sure you get all the questions. They went up so fast. Let me see. Okay, here we go. Again, the concept of fermentation is the idea that you need a way to keep glycolysis going, and what you need to do is you need to remake NAD+. So fermentation is like an alternate pathway to get you there, since you can't do the ETC. What is the 4-carbon molecule that is produced during the Krebs cycle called? You really don't need to know about that molecule. I wouldn't worry about it. I believe it's called oxaloacetate. It's okay. Here you go. DNA overload is the idea that if you have a really big cell, you just can't make all the proteins it needs with that one set of DNA, right? It would be like trying to use one cookbook to cook enough food for like the city of Los Angeles. It wouldn't be possible. Well, we have to know stuff like, you need to know the palisade mesophyll. I didn't talk about the cuticle. I didn't talk about the epidermis, so no. When there is no oxygen fermentation, yes. Okay.
I can't wait.
Let's see. So does all of cellular respiration? No. Glycolysis happens in the cytoplasm, but then the rest of cellular respiration, which would be the Krebs cycle and the ETC, happens in the mitochondria. Let's see. Citric acid is actually the 6-carbon molecule that you form when you join acetyl-CoA and that 4-carbon molecule. But you don't need to know that, I don't think, or at least my students don't. Let's see. Is the Krebs cycle... In the Krebs cycle, does pyruvic acid lose? Yes. In the Krebs cycle, the first thing that happens is pyruvic acid loses the carbon dioxide. It comes out as CO2, and then CoA attaches to CO2. You want this? Okay. Okay.
We have a pen in there.
Yeah, a pencil, huh? Okay. Let's see. In Krebs cycle, after 3-carbon, where does one of the carbons literally leaves as CO2, like I said, and becomes acetyl-CoA. How does a stem cell form? A stem cell doesn't form; it's already there, and the idea is it just slowly divides and stays and gets ready to.
What a reaper.
I don't know, baby.
What is it?
I don't know. Let's see.
What a reaper.
I don't know, baby. Sorry.
Where is it?
Oh, the eraser?
No. No.
Let's see. Is ATP made in the Krebs cycle, or the majority of it is made in the ETC. A tiny, tiny bit is made in the Krebs cycle, like very tiny, like 2 ATPs. Which part of mitosis is spindles form? And spindles form in prophase. Does transpiration involve cohesion, adhesion? Technically, transpiration is just the evaporation of water. Like, that's what transpiration is. But as sort of a collective process of how you then draw water up the xylem because of transpiration is a combination of cohesion and adhesion. Okay, Marissa, how are interphase and mitosis related to prophase, metaphase, interphase? Okay, so interphase is all the stuff. Oh, hold on, baby. Interphase is all the stuff like that happens before prophase, metaphase, anaphase, telophase. Mitosis is actually all of that stuff I just talked about. Like mitosis is prophase, metaphase, anaphase, telophase. That is mitosis. Okay. Can I talk about fermentation? Yes, I'm not sure quite what you want me to say, so I will try again to review. Fermentation, again, remember, is going to be the, it's basically going to be how you regenerate NAD plus in order to keep it glycolysis going. Let's see. During transpiration, is water pulled up like a chain by cohesion? Yes, but well, so basically, again, it's sort of like a chain reaction, right? That transpiration is the evaporation of water from the stomata. When it evaporates, you have now like an empty space in the leaf. So now the water at the top of the xylem moves in to fill its space, and because that water moves up, all the other waters get pulled along because of cohesion and adhesion, etc. Which then, yes, all plays into supplying the plant with water. The ETC, again, is just a bunch of proteins. You pass the electrons down it. Ultimately, it lands at some sort of final electron acceptor. So in cellular respiration, the final electron acceptor is oxygen, which then makes water. In photosynthesis, the light reactions, the final electron acceptor would be NADP+, which then makes NADPH. Yes, since each cell has 46... What do you want?
Mommy, move.
What do you want?
Mommy, move your computer.
I can't move my computer. I need to use it. What do you want? Not this one? Oh, hold on.
Move it.
I can't move it right now, baby. What do you want me to do? What do you want? Okay, I can't do that. Oh, hold on. Okay, let's try this. Oh, okay. I'll try it this way. Let's see. Does the sunlight absorbed in the palisade mesocycle somehow transfer? No, it's just that the sunlight is able to get past that, which it can. Then it would get to the sun. I mean, I suppose some photons of light could bounce off of the cells and not enter them. But yes, if each cell has 46 chromosomes, you do have, yes, trillions of chromosomes in your body total. A primary consumer is a plant. A centromere is basically where the two sister chromatids attach to each other. Do all plant cells have chloroplasts? No, they actually don't. The cells in the plant that are photosynthetic have chloroplasts, but there are parts of your plant cells that don't have chloroplasts. But for our purposes, again, we don't tend to do that. Are the 3 limiting factors surface area development ratio? Yes, that is for limiting factors of why cells need to be so small. When do we perform lactic acid fermentation? Why? You perform lactic acid fermentation when you don't have oxygen. Now, remember, that means a particular cell doesn't have oxygen. It doesn't mean like your whole body doesn't have oxygen, right? So you get your oxygen through your lungs, but even if you breathe in that oxygen and the lungs have the oxygen, now they have to transport the oxygen in your blood to all of your cells, and sometimes you just can't get there fast enough. So that means, let's say you're running the mile or you're swimming like 70 laps, then at some point your cells might be not able to get oxygen fast enough to keep up with the cellular respiration they're doing. So they would switch to lactic acid fermentation temporarily so they could still generate some energy until they get the energy oxygen, but it's temporary. Let's see. Why do we produce more CO2 when we exercise? Again, when you exercise, you're doing more cellular respiration because you're going to essentially need ATP for your muscles to contract, if you want to sort of be very specific. So because of that, right, you're doing more cellular respiration, and CO2 is a byproduct, the waste product of cellular respiration. So that's why you make more CO2 when you exercise, because you're doing more cellular respiration. So the reactions are happening more frequently. Why are we often told we have 23 chromosomes? You don't have 23 chromosomes. You have 46 chromosomes. You get 23 from mom, 23 from dad. Catabolic and anabolic usually refers to types of like processes, like either like usually molecules that like build stuff up or break things down. My students, you don't need to worry about that. Two sister chromatids are, yes, two sister chromatids are referred to as a chromosome after replication. Okay, thank you. Okay, thank you. Okay. Oh, okay. I have to talk to mommy's students. You want to say hi? Okay, then maybe you should go outside and see if daddy's watching a show. Okay. Okay. Sorry. Okay. If there's glucose in all the food we eat, so again, not all food has glucose. All food has some sort of biomolecule which you can churn into something, which will then be made into some part of cellular respiration.
I wanna Hannah.
No, I need you to go brush your teeth, please.
Hannah.
What is it you want?
I wanna
Why is glucose from certain foods better? Well, so that has more to do with nutrition, but it's the idea of like processed sugar versus like natural sugars and things like that. Yes, aerobic means it requires oxygen. Anaerobic means it does not require oxygen. Cohesion and adhesion have to do with basically they're different because cohesion is water sticking to other water molecules, and adhesion is water sticking to other water molecules.
Tell them wait, who were those students who were pretending? What's their names?
Oh, I don't know their names, sweetie. And I can't talk to them right now, sweetie. Okay.
Alicia. Their
No,
names are
no, don't go making things up.
Didn't Daddy, Mommy tell them about the problem that happened?
Okay, I'll tell them later. I need to finish answering their questions. No, I have to finish doing this first. Thank you. Go outside, please. So what causes? No, you don't need to know about meiosis. Oh my, it went upstairs. Okay. Cancer occurs essentially when cells have developed enough mutations, enough mistakes in the DNA that they start to not listen to the signals that tell them when to divide and not divide. Again, stem cells are undifferentiated cells. They're like precursor cells that could become anything. Think of them as like, no, thank you, Sammy.
Tea.
No, thank you. I don't want you to spell that. No, that's not tea. Thank you. So think of stem cells as like dough that you can still make into any kind of bread, right? You could there's lots of different kinds you can make a bread, like lots of different kinds of bread you can make, and stem cells are like that raw dough that still could become any of them. Let's see. How is what related to transpiration? I'm not sure. Cholesterol. Okay, cholesterol. Let's see. Was it Ria who asked what? Okay, the function of cholesterol is in the cell membrane. It actually keeps your cell membranes from not being too liquidy. So that's what it does. It's not a phospholipid. It's actually a lipid, but it's not a phospholipid. No, thank you, Sammy. Thank you. No, don't push buttons. Thank you.
No.
Okay, you almost did. Thank you. Why do cells? Well, because cells don't necessarily have to reproduce more cells. If you just kept dividing cells for no reason, you would just have all these extra cells in your body. So cells only reproduce when they're needed to reproduce. What exactly? Again, P53 is it's like a proofreader.
This is a knife.
Oh, please don't play with that, Sammy. That's a pencil sharpener. P53 doesn't produce the cyclones. P53 sort of gives the go-ahead for cyclones to be produced. You sharpen a crayon, but please be careful, okay? Mutations happen basically when the DNA is being copied. There are mistakes that happen.
You're gonna ruin that.
Okay, go outside, please. Thank you.
Huggy,
Bye-bye.
kiss, hug,
Okay,
kiss.
hug and kiss.
Hug.
Hug, kiss. Okay, go. With daddy. Thank you. So mutations happen basically when in that process of the chromosomes copying, mistakes happen, right? Imagine if like you were given like 40 pages of a book you had to copy, you might make some mistakes in there, and that's essentially how they happen. Similarities and difference between photosynthesis and cell respiration. The similarities are that they both involve ETCs, they both involve energy molecules, they'll have energy carriers. Obviously, the differences are very large. They make different things. They're actually kind of almost like opposite, reactants and products and things. Um, let's see. Let's see. Uh-oh. Um, the test again will be mostly multiple choice, little tiny bit of, mostly multiple choice, and then one A level question and some really short, like short answer, like very, very short. Um, how difficult? I think it depends on how well you understand the material, honestly. It's not meant to be tricky, but it could be harder. It could be easier depending on how you're feeling about cell respiration and photosynthesis and things. Um, during photosynthesis, when does oxygen leave the stomata and when is it used? It really depends. Like, remember, the stomata are not controlling anything. It's just like an open window. So if there's oxygen, it could flow it out of the stomata, but it might not if it's also just being used up by the cell in photosynthesis. Or sorry, cellular respiration. How does potassium affect the closing? Again, potassium is the molecule that causes the stomata to open or close. You pump potassium out, water follows, stomata close. Pump potassium in, water follows, stomata open. Does minerals get into the roots by osmosis? No, minerals get in the roots by active transport, and then it goes water follows by osmosis. So really, what's in the xylem is a combination of water and minerals. But no, things that are going up the xylem are just floating up, like they're going up by cohesion and adhesion, and then the minerals are kind of mixed in there. No, cells pretty much just self-destruct through apoptosis or necrosis. My students, we didn't talk about necrosis. Don't worry about that. The ETC again is a series of proteins where you're dropping off the electron, it passes it down the chain, and then at the end you have to give that electron to some sort of final electron acceptor, and in the process you actually generate ATP because part of that process turns ADP into ATP. Yes, okay. All right, it says osmosis is diffusion of water. Okay, yeah. Okay, let's see. Okay. Yeah. So oxygen, the oxygen can either, if you break apart water and you make oxygen, right? That one oxygen, yes, can join with another oxygen to generate oxygen gas, which then could go to cellular respiration and be used as a final electron acceptor, but it could also theoretically just float out of the stomata. It just depends. Um, so again, so good question. We don't have chloroplasts, right? We do not have the ability to go out into the sunlight and transform the sunlight's energy into glucose. That's why for us, since we are heterotrophs, we have to eat food, and then the food we eat then gets broken down into biomolecules, converted into some form that's either glucose or something very similar to glucose, and that's able to then go into cellular respiration, and then that makes ATP for us from the glucose that we eat. Okay. Any other questions then before we go? It looks like we're almost out of time here. So if any last questions, then I can go over them now. I think I got everything. If I missed your question, please do tell me because the thing kind of jumped around really fast. And so I think I tried to catch up from where I was, but I wasn't sure if I got anything, if I missed anything. As always, you're welcome, guys. As always, if you have questions tonight, you can email me. You can come and find me tomorrow. You're welcome. During tutorial, I'm in 105. How can you remember the equations? Honestly, I would just try to remember what goes in and out. It's one of those few things you're going to have to memorize. Um, but the good thing also you can remember is like the photosynthesis and cytosolution equations. They're like opposites of each other, right? So if you remember one, you just have to reverse them to remember the other one. So yeah, again, email me tonight if you have questions. Oh, how does water doesn't go into the stomata. That's actually a really important point. Water does not go in the stomata. Water only goes up the xylem and then can go into the leaf space of the leaf, but it actually never goes in the stomata. So like um it's a common misconception, right? Water only evaporates out of the stomata, it doesn't go in. So it's really important that you don't accidentally think it doesn't drop in through the stomata. You would think so, but it doesn't. Guard cells are not made of stem cells. No, guard cells are specialized cells in the leaf whose job it is um, to, you know, open and close the stomata. Yes, so water enters a plant only through the roots. So, like, if somebody like mists their plant, they're not actually watering their plant. What they're doing is they're creating like a humid area around the plant, and that's going to let the plant not transpire so much, so it doesn't close its stomata. So if there's water in the air, right, then that's water vapor that helps sort of balance how much transpiration happens out of the stomata, but it doesn't drop in. Now, if there's no water in the soil, that's when your plant basically starts to shrivel up and die because you can't you're still using the water, right? The cells in all of plant cells are using water. Photosynthesis is trying to use it for photolysis. So if there's no water in your plant, in the soil, that's why your plant will start to wilt because it's using water but not getting any water. And basically, that's all limp. Any other questions? Okay. So, my students, remember you have the other resources also posted in Google Classroom if you need those. Hopefully everybody can see those. If you have questions, send me emails tonight. I will be online tonight. And sorry again for the extra people in the room. My children really get a kick out of looking at the videos and stuff. So, anyways, so thank you for your patience with them. All right. Have a good night. Remember, get a good night's sleep. Eat a good breakfast tomorrow morning, um, so that you have, you know, well rested minds for your test. Okay. All right. Bye, guys. Oh, I saw one quick question. So it says, are stem cells in both animal cells? Yes, stem cells just mean a cell that could become a future cell, but hasn't gotten its assignment yet. So yes, you can have stem cells in plants or animals. Okay. Bye, guys. You're welcome.